Biochemistry at University of Texas Medical Branch in Galveston. He's been researching. He's basically a biophysical expert, so you can ask him all the questions with regard to the interaction of small molecules and proteins with macromolecules. But okay, what happened? He disappeared. Researching glycosaminoglycan binding to proteins, and it so happened that I looked him up, and he applied for a big award, a PO1 award. So he was part of that PO1 award, just as Kuberan Balgurunathan was also part of the PO1 award. And so the program project has ended, but we continue our interaction. And he has graciously agreed to give these 2 hours. So ask him any questions that you would like to, because this topic directly relates to what many of you might actually do in the lab. Okay, Krishna, all yours.
Thank you, Umesh. Good afternoon, everybody. So Umesh had asked me to talk to you guys about, in 2 hours, 2 classes, aspects of protein-GAG interactions, in particular thermodynamics, kinetics, and dynamics of protein-GAG complexes. Okay, so I'll try to do the best I can. So, feel free to stop me if you have any questions, and I'll try to do the best I can. Okay, so I think the last few classes you guys were introduced to about the structures. Okay, so at the end of the day, all of us are interested in trying to understand, whatever we're studying, what they do. I mean, how they do what they do. Like, for example, in the context of GAGs, we are interested in how proteins bind to GAG. Okay, to be able to get that information, we need the structure. Okay, so but once we get the structure is just a place to start. Okay, so we are also interested in going from there, how it binds, where it binds, and so on and so forth. So let me expand upon that. Okay, so so that's sort of my next slide shows, to be able to understand the molecular basis of protein-GAG interaction. We need to understand the relationship between the structure, the dynamics. So I'll sort of come very soon. What do I mean by dynamics? Kinetics. So kinetics is a sort of a manifestation of dynamics, and thermodynamics. Okay, the thermodynamics is central to anything and everything. And once again, as the talk progresses, hopefully it will be clear where we are going with this. Okay, so all of you in the last few classes, and considering all of you work on GAGs, I think I need to spend the least amount of time introducing the GAG structures. Okay, so the GAG structures we are interested in is the sulfated GAGs, in particular heparan sulfate. Of course, all of us use heparin as a surrogate for understanding heparan sulfate, and also less well studied. It doesn't mean less important. GAGs like chondroitin and dermatan sulfate. So you guys know they are linear polysaccharides and repeating disaccharide units, and the challenge, of course, when it comes to in vitro and in vivo, this intrinsic heterogeneity in terms of NNO sulfation and so on and so forth. Okay, so like in the next 2, I sort of talk about the system I'm going to be discussing, I talk about gradients and modulating monomer dimer equilibrium. Okay, let's discard that for time being. Okay, so once again, as I alluded to earlier, so what are the most pressing questions that limit our understanding of how GAGs bind proteins? Okay, and the fact remains that the molecular mechanisms and the structural basis are lacking. We know very little of how GAGs interact with proteins, and even that for very few proteins, like for thrombin and maybe a couple of proteases. Okay, so intrinsic to that information is then in general for GAGs. Okay, so what are the binding affinities? It depends upon the length of the GAG you use, the nature of the GAG you use, the technique you use. You can get very different answers. The stoichiometry, okay, for example, how many proteins are binding to a single GAG? As you know, GAGs are long polysaccharides. So depending upon the length, you can get different stoichiometries. And if you take as a defined length, okay, depending upon the technique, you can come up with different numbers. And there are not many techniques which will give you very stringent stoichiometry. And the kinetics, basically the rate at which the protein and the GAG come together to find a complex, and the rate at which they dissociate, so to speak. And thermodynamics is, basically is relationship in anything and everything. I'll come to that soon too. Okay, so so why are we interested in all these things? Okay, so almost all GAG binding proteins exist in multiple forms, like monomers and dimers, and almost invariably all proteins dimerize on GAG binding or vice versa, like growth factors, chemokines, cytokines. Okay. So what exactly is the relevance of these interactions? And to zero in terms of function, what are the GAG binding residues? Which residues are exactly binding, and how their binding dictates affinity, specificity, and the geometry. Okay, so how do we gain insights into structure, dynamics, and thermodynamics supporting GAG complexes? Okay, so in other words, what are the experimental methods which will allow you to answer these questions? Okay, in addition to experimental and the computational methods, we will see very soon. A lot of this information can be obtained only from computational methods, and there are no experimental methods. And once again, I'm harping the same thing over and over again. And why is it so? Why is this so knowledge so hard to come by, despite their importance? Okay, so why study and what is thermodynamics? Okay, so thermodynamics is intimately linked into all these different laundry list kind of things I've plugged in here. How does the structural features mediate binding? The structural features could be that of the protein and of the glycoside and glycan. What is the binding mode? Okay, so how does it bind? Meaning the binding geometry, which residues are dictate the binding interactions, and why these residues? Okay, so I guess when you guys were studying on the structural aspects, I think you were introduced to the interaction is mediated predominantly by basic residues like lysines and arginines and histidines. Okay, why these residues? And also, I think there was some discussion about the polar residues too. Okay, so what is the contribution of the individual residues? If there are 5 lysines in the binding site, do all of the 5 lysines play a similar role, or some residues are more important than the others? Okay, things of that nature. Are the binding interactions additive or cooperative? For example, if there are 5 lysines and the binding energy is 10 kilocals, okay, so does the individual lysine each contribute 2 kilocals? 2 times 5, 10. 1 or 2 lysines are basically responsible, contributing to 7 to 9 kilocals, and others are just 1 or 2 kilocals. Okay, so if you do a mutation, if you mutate one lysine to an alanine, and if you see the change in the binding energy is just 2 kilocals, okay, so you do the individual mutations, do they all have to add up to 10 kilocals, or it could be 8 kilocals or 15 kilocals? Okay, in other words, the additive or cooperative is there crosstalk between the different lysines. And so what is the relationship between the kinetics? Okay, so kinetics in terms of, as I said, the on constant and the off constant. And the thermodynamics is basically, I'll come to that, it's the binding affinity. Okay, so, and why is that we need to understand the kinetics for understanding function? Okay, so I'm sort of repeating the kinetics and thermodynamics and the dynamics and thermodynamics. Okay, so any questions so far? I'm just reading off a whole bunch of what I feel is really background information. And I'll sort of give specific examples and sort of try to bring in the big picture. Any questions from anybody before I go forward? Because I can't see your face, it's very hard for me to gauge what exactly is happening on the other side.
No questions here.
Okay, the same thing elsewhere. VCU, no questions from Wisconsin. So far, so good?
No questions from Wisconsin so far.
Okay, great. So are you guys able to hear me clearly?
Yeah, sounds good.
Okay, so no issues.
Yep.
All right. Okay, all right. Okay, so what are the challenges or factors that needs to be considered for studying thermodynamics? Okay, so basic knowledge we need is the structures, or like structural models, and intrinsic to that is the limitation of the structural techniques, or biophysical techniques, as the case may be, the complexity of the binding interactions, and the complexity of the naturally occurring GAGs, and the non-availability of homogeneous GAGs. Okay. So yeah, a very important aspect of GAG interactions is the specificity versus chemistry. Okay. So are GAG interactions specific? This is especially relevant, like for things like epron sulfate, which has large diversity in chemical structure. Okay, so does that play a role in terms of whatever it does? Okay, so how do we define specificity? How do you measure specificity? Okay, so and that's once again intrinsically, coupled to the thermodynamics. And so also purely from an evolutionary perspective, because the GAGs are there everywhere, and how the protein of interest have evolved to bind a specific chemical structure of the GAG, or like epron sulfate, and the intrinsic diversity of the GAG structures are able to accommodate all different proteins they bind. So what does a measurement of a binding affinity mean? How does that relate to function? For example, if a particular protein binds a particular sequence, GAG sequence, particular chemical structure with higher affinity, does it mean that it has higher specificity? How do we even characterize specificity? Okay. Okay. So in other words, coming to the other chemical level, what interactions mediate binding? So in terms of what the measurables, okay, so what we know is our textbook description is that the interactions are mediated by electrostatics. Okay, so the GAGs are the most highly negatively charged biomolecules, with they happen having 2 or 3 sulfates plus 1 carboxylate. Okay, so not surprisingly, if you look into the literature, the most studied, like 99% of the literature, or whatever protein you look into, will talk about the electrostatics. Okay, so the residues which are most highly charged basic residues are the lysines and the arginines. Once again, I think you guys got an introduction to this, and Martin talked about the structural basis of GAG interactions. Okay, so another residue of interest is histidine, which is basic. It's basic, but it doesn't have the same properties as the lysines and the arginines. So I'll sort of spend some time talking about the characteristics of these residues and what does it mean in terms of structure and function. Okay, so now, so what exactly is thermodynamics? Okay, so it's best described by these 2 equations. Okay, this is textbook description. Most of you guys got over the first taste of this, like in high school, or when you're taking physical chemistry, or like biochemistry. Okay, so delta G is, which is the Gibbs free energy, equals to delta H, which is the enthalpy changes, minus T times delta H, which is the entropy. Okay, delta G is also related to ln K, where K is the equilibrium constant. In other words, delta G is nothing but the equilibrium constant, the binding affinity, so to speak, and which is a function of 2 variables, which is defined in a stringent fashion using the delta H and the delta S terms. Okay, so this is just a bunch of numbers, right? You do an experiment, you get delta G, delta H, and delta S, or whatever it is. So how do we interpret these numbers in terms of what we are interested in? In other words, how GAGs do what they do, and to be more precise, how do GAGs bind to proteins? Okay, and as I alluded to earlier, I'll keep coming back over and over and again. We need structures or structural models to be able to provide this sort of a description of how the binding interactions mediate and give the final binding affinity. Okay, so when the enthalpy, okay, so is generally interpreted in terms of favorable packing interactions. The packing interactions could be like hydrophobic interactions, like the large hydrophobes packed against each other. These are much better, well described for like protein-protein interactions, and maybe even like protein-nucleotide interactions, but like not that much for GAG interactions. And things which are more intuitive is the ionic and hydrogen bonding interactions. Okay, so these are generally we think of enthalpy in terms of favorable interactions which promote binding. Okay, the more favorable these interactions, the tighter the binding. Okay, it's intuitive. And so once again, the delta H is the favorable interactions are minus. Okay, so the delta G is also minus. So the delta G is like minus 7 kilocalories. That means binding is favorable. Okay, so once again, I would like to say that right up front. And we need the structure. So sort of the enthalpy, we can look at it as like when you have like two molecules, the protein and the GAG, which are sort of flexible, dynamic, rowdy. They are just all over the place. But when they bind to each other, they are more structured. They are more well behaved. So more ordered the system, the higher the enthalpy. Okay, sort of get like a qualitative description. So the other variable is the entropy. So the entropy is dictated by the dynamics. Okay, so all biomolecules are highly dynamic. Okay, so the dynamics is absolutely critical for function. If they're not dynamic, they cannot bind their partner. There's no function period, and the dynamics plays very important role in recognition and affinity and specificity and so on and so forth. Okay, so I said like that we can simply look at dynamics as motions. Okay, so like you have the methyl groups going, rotating the like helices and the strands, like segmental motions, and whenever there's a bond, there is motion. And these motions, most importantly, come in different time scales. Okay, so like motions in a nanosecond, picosecond time scale. So a picosecond is 10 power minus 12. That means whatever motions we're talking about, it happens 10 power 12 times per second. It's extremely rapid. So there are motions at very, very fast time scales, nanosecond, picosecond, and like millisecond, microsecond time scales. Okay, 10 power 3 to 10 power 6 per second. A lot of the motions which are biologically relevant are these what is called slower motions, like millisecond, microsecond time scales, and motions which are much, much slower, like seconds and times and hours and so forth. A lot like this helical motions and strand, like when you have like large segments moving, they can happen at much smaller scale. And the tumbling of the molecule itself happens like a nanosecond, picosecond. So that is like the dynamics of the protein. In general, when the binding occurs, because of restricted motion, the molecules tend to be less dynamic in the bond form compared to the free form. In addition to that, entropy is intimately linked to the organization of the water, okay, because we're talking about molecules which are highly negatively charged opposite to each other, as the case may be. There's a large number of molecules which are restricted around these molecules, around the GAG or the protein. So, but when they come together, right, especially the binding interface, these water molecules are released. The release of water molecules increases the entropy of the interactions. In a similar fashion, there's also release of cations like such sodium and so on and so forth. So entropy is we understand like the basic principles, but trying to really understand all of the entropic changes is very, very challenging. Okay, so at this stage, basically, I think where we are is just trying to be able to appreciate whether the binding is enthalpically driven or entropically driven or a combination of both. It is in itself, we can get that kind of information. I think it will be quite useful. I think I'll show you once again a couple of examples with respect to that. Okay, so so we already I already alluded to this. Okay, the role of ionic interactions. So the ionic interactions could be short range or long range by its very nature, and so we'll sort of talk about that in the case of lysines and arginines, and I will sort of also give examples of the non-ionic interactions, how much they could contribute, and what exactly dictates these interactions, and so on and so forth. Okay, so so what exactly are the residues which mediate ionic interactions? Okay, it's basically lysines and arginines, and here I'm showing the side chain structures. Okay, so the lysine over here, okay, we have a beta, gamma, delta, epsilon. So it has a really long side chain at the end of which you have the NH3 group, which interacts with sulfate and the carboxylates. Okay, so in the case of arginine, we have like 3 carbons, but its side chain is a lot more complicated. Okay, whereas ammonium is really simple. This side chain is a lot more complicated. You have like 4 nitrogens and an epsilon H, and so it's more asymmetric. So the nature of these interactions, people who are chemically inclined, and so you can appreciate right away the nature of the interaction, the binding affinities, and everything is going to be very different from lysines and arginines. Okay, so but a lot of the literature basically couple lysines and arginines together. So we sort of spend some time looking at the differences and how they mediate binding interactions and affinity and specificity and so on and so forth. The histidine, once again, it's a very unique amino acid. Okay, so it is side chain. You have the imidazole ring, which has 2 nitrogens. Okay, so which either of them could be protonated. Okay, and that's also whereas the pKa of these side groups are like more than 10, this tends to be around like 5 or 6, which is more physiological. And much less histidine is much less well studied. Okay, so there are very few examples in the literature where people have shown direct involvement of histidine. So clearly, because of the side chain structure, it can play specific roles. And quite a few of GAG binding proteins do have histidine as one of the conserved residues. Okay, so how do we study ionic interactions? Okay, so like all of the data I'm going to be showing you today in terms of getting across these interactions are spectroscopy, nuclear magnetic resonance spectroscopy, and of course computational. Okay, there's something Dr. Desai is a world expert and has developed quite a few methodologies to study these interactions, and I'm also going to give specific examples. So is there a question?
Nope.
Okay, cool.
No questions yet.
Okay, all right. So in addition to ionic interactions, we could we also have, once again, as I said earlier, the weaker interactions, which is basically hydrogen bonding interactions mediated by polar residues like asparagine and glutamine, and that should be a serine, and I is missing, and much less well studied. But I'll sort of give examples of how we can study them. Once again, the techniques which are available right now are in terms of direct characterization and solution NMR and like molecular dynamics. In principle, you can do like mutations. As far as I know, I don't think there are any published literature where people have actually mutated experimentally an asparagine or a glutamine and shown that it's involved in binding. I think that's simply because I think the binding energies are really small, and I think that at this time we don't even know. There are very few examples of bona fide asparagines and glutamines playing a role in GAG interactions and mediating affinity or the specificity, as the case may be. Okay, all right, I think we already saw this. Okay, so coming to the methodology, okay, so this is where I think the challenge and the limitations are. Okay, so we are, you want to study binding affinity, you want to understand what is the stoichiometry, you want to study the kinetics or thermodynamics. So by the end of the day, you are limited by what techniques are available, what tools, what are available in your lab or like in a core facility, and how easy or how challenging are these experiments. The simplest experiments, which almost all of you will be familiar with, like fluorescence based, okay, because I think most labs will have a spectrometer or have an axis. A lot of times you measure anisotropy or like an intensity, and like you can also do something like a CD where once again, you need to have an access to a CD instrument. You can get some kind of binding interactions, okay? But they are these like fluorescence-based and CD-based studies are, at the most, you can get intensity. They do not have any structural information. Definitely, you cannot study thermodynamics with fluorescence. Maybe you can get some insight into kinetics, okay, depending upon the label and things of that nature. The technique, default technique we think of to measure thermodynamics is isothermal titration calorimetry. So I'll come to that. And to measure kinetics, the on constant and the off constant is an SPR. SPR is these days common, like generally any major university will have like an SPR either as a part of the core or like any labs. And techniques which can give you information of the binding of like the structure of the complex, like crystallography and nuclear magnetic resonance spectroscopy. And so crystallography, the challenge is invariably the protein sugar complexes will not crystallize. And if they crystallize, they will not give you good diffraction and so on and so forth. If you were to go, if you're a structural person, if you were to go to like the protein data bank, which has all the structures, you'll see like the protein GAG structures are like less than 1%. I think it's more like 0.2% or something like that. Very, very few structures. Even that of very few proteins. Giving you some kind of an idea of what challenges are there to be able to understand at a molecular level what's happening. And something which is easier and more straightforward to do is like molecular dynamics and modeling. You can do it if you have a structure as well and good. Even if you don't have it, if there's some functional data or even like from first principles, you can do like MDN modeling. Okay, so the techniques I'll be sort of alluding today would be the ones in yellow. So I'll be talking about ITC. In fact, I think that's where we'll be going very soon. I'll also talk about SPR for kinetics and NMR and the modeling. Okay. Okay, so any questions so far?
No, sir.
So you guys have understood everything I've said so far?
What did you say?
So I might assume that everything I've said so far has been crystal clear. We are all on the same page?
Yes, sir. Yeah. It's all very organized and clear.
Okay, excellent. Okay, all right. So now we will come to the technique. Okay, so the technique of choice is isothermal titration calorimetry, or ITC, the short form. So like in an ITC experiment, basically a single experiment will give you information on the affinity or the delta G and its thermodynamic parameters, enthalpy and entropy, and stoichiometry, I think, which is generally it's not a big deal for for most systems, protein, DNA, RNA, you know. But for GAGs, it's challenging. You really don't know how many proteins are binding per GAG or vice versa. So in that sense, ITC is very useful. Okay, and all this can be done using a single titration. Okay. So as I said, the reason why ITC can be very useful is it can give you the relationship between stoichiometry and affinity and the GAG size. A lot of the times, for working with different other biomolecules, size doesn't come into a picture. Okay, but with respect to GAGs, size definitely comes into picture. Just to give you a historical perspective, a lot of, if you were to do a literature survey, a lot of structural studies or biophysical studies, people tend to use very small size GAGs. The smallest you can go is like a disaccharide, okay, maybe a tetrasaccharide. And that's simply because if you go to a longer oligosaccharide, even like an octosaccharide, the protein scrap or the complex scraps out, which means you can't study them, simply because of the nature of the biomolecules. But ideally, in vivo, the GAGs are much, much longer, right? So they're polysaccharides. So the longer they are, they reflect more of the reality of how the binding occurs. So the size definitely makes a difference, and we will, once again, I'll give you examples of that. And for historical reasons and practical reasons, most people, including us, we study heparin and not heparan sulfate, though physiologically the proteins of interest bind to heparan sulfate. But heparan sulfate is modular in nature, and also intrinsically the heparan sulfate, there's quite a bit of diversity. So that's because that heterogeneity, it's not an ideal candidate to study. On the other hand, heparin is more uniformly sulfated, and you can commercially get different heparins of different sizes. So it's convenient to be able to do biophysics and structural biology, but at the same time, you always need to worry how much of whatever you're studying using heparin reflects binding to heparan sulfate. Okay, so what, in addition to enthalpy and entropy and things of nature, you can also measure things like heat capacity. This is pure physical chemistry, so don't sweat. The more we know, the better it is. So I'll sort of give you an example of what is heat capacity and what it is telling us when I come to the data. And so what advantages do we have with ITC? There are no modifications, okay, I think which is pretty cool, because if you're doing fluorescence, you have to put a fluorescent tag, because GAGs, they do not fluoresce. Okay, generally you put a fluorescence tag, or like a lot of folks use the intrinsic fluorescence of the tryptophan of proteins and change in tryptophan fluorescence as a readout for measuring binding. But the change in tryptophan fluorescence is really small, and it's really not, in my opinion, a reliable measure for binding. There are papers in literature where people using tryptophan fluorescence talk about binding affinities like a nanomolar for like a disaccharide and so on and so forth, and you use other techniques and the binding is actually millimolar or different binding at all. So there are a lot of artifacts simply because of the nature of the technique. So using tryptophan fluorescence, the intrinsic tryptophan fluorescence, in my opinion, is not a good choice. So, and so that it's it sort of comes back to what I've been alluding to earlier. So like techniques-wise, there aren't that many techniques which are friendly to characterize GAG interactions. Okay, so another advantage is you measure in solution. Okay, unlike in immobilization, like for example SPR, so immobilization can introduce artifacts, and of course things can get more complicated. And as a general introduction to ITC, what's great about ITC is also it's the large differences in binding constant. You can measure binding if it's as low as millimolar, as high as nanomolar. Like for example, fluorescence, you cannot measure by very high affinities. Okay, it has to be like nanomolar, low, or micromolar. And depending upon the system, it can also get mechanistic insights like cooperativity and things of that nature. Okay, so from a very practical perspective, considering this is a medicinal chemistry course, right? And I'm sure quite a few of you are like pharmacy students, and a lot of you may want to end up going to work for big pharma. And I'm a big proponent of ITC, simply because of what it does, and I think it's very important from a drug development perspective too. So I'm sort of going to talk about that. So like at the end of the day, whatever system you're working on, from a drug discovery perspective, we're always interested in high affinity and high specificity inhibitors. So why knowing enthalpy and entropy contribution important? Okay, so because a lot of high throughput screening methods, like even computational docking, pays attention only to the enthalpic factors, so-called the binding interactions, because it's very hard to model entropy issues. So you can have very high affinity binding. Remember that delta G is delta H minus T delta S. So both of them contribute to binding. So the binding is a function of both enthalpy and entropy. And so if you can have tight binding and in the binding pocket, in the bound form, the protein and other complex continue to be dynamic. So the binding is driven both by enthalpic and entropic factors. Okay, so as I said, entropy is difficult to model. Okay, so as I said, also like the scoring function for entropy is very weak. It's completely inconsiderate. And these days you can get high throughput ITC, like second, third level screening, to sort of zero in, where you want to go from micromolar to like sub-micromolar to a nanomolar, which is the challenging part. Okay, of course the challenge is you need a lot more protein than traditional high throughput screening approaches. But technique is being automated, miniaturized, and making it more approachable. Okay, so ITC is good. Okay, so the nuts and bolts. So this is sort of the math below the how we get whatever we get. Basically, what we measure is the heat, and which is related to, the binding constant and the protein concentration and things and so on and so forth. Okay, just FYI, just for information. Okay, so the instrumentation is, it's come a long way. Basically, it's a desktop, and it's like you can get like one for $125,000. It's not even that expensive, relatively speaking, of course. And so you have like a reference sample, and what you really are measuring is very small changes in the heat. Okay, that is your readout. Okay, and from I'll skip the details. So basically, from the change in the heat, if you remember the math I showed you, you can get all the binding information. So this is how the ITC output looks like. Okay, so you start adding, and you're basically measuring the heat release as you're adding, for example, the GAG to the protein. Okay, so then as the binding is saturated, there's no more heat release. So when you integrate the heat, right, and this is what you get, this kind of a profile. So the y-axis is proportional to the heat released, which is sort of proportional to the enthalpy. The slope is related to the affinity, and the inflection point is sort of related to the stoichiometry. Okay. Okay, so we already talked about this. So the disadvantages are you do need a lot of protein and GAGs are not cheap. Okay, so if you're commercially buying or if you're making it in the lab. So, and having said that, depending upon the kind of questions you're asking, and if the heat release is really high, right, you need very little protein on the GAG, and you do need some expertise. Okay, it's not, and you need to be extremely careful when you're doing the experiment because of the precipitation issues and things of that nature. But it's very doable, and I think the information content is very high. Okay, so now I'm going to give you some real life examples of where ITC was used to gain insights into the thermodynamics. Okay, so these are the proteins we work in the lab. So we work on a family of proteins called chemokines, which recruit neutrophils. And humans have these 7 different chemokines, and there are also 2 mouse chemokines. Okay, so we have done quite a bit of work trying to understand how these chemokines bind GAGs, in particular heparin. Now, what we see is that there are some residues, like for example, which are highly conserved. Okay, like for example, B3 you can see, and like B8 and or the B1, but some are not that conserved. Like some have and some don't. And what I'm showing here in blue are residues which are unique to a given chemokine, which are involved in GAGs. So if you look at the cells, the structures of all chemokines are similar, but if you look at the charge distribution, the red means they're negatively charged, blue means positively charged. You see the different, the topology, the electrostatic distribution is not the same, though it seems when you look at the individual sequences, a lot of the residues look alike. Okay, so here's sort of the summary of which residues are conserved and which residues are not conserved among the related chemokines. Okay, so how does that translate to the binding, right? So here I'm showing actual data to the mouse chemokines. This is the work we did recently with Dr. Desai, which was published last year. Okay, so we are taking 2 mouse chemokines. These chemokines are very well characterized in mouse inflammation and pathology, and like in any disease models, these are the chemokines people talk about. So this is called MIP2 and KC. And so one thing we know is that the dimers bind with higher affinity, and the dimerization and GAG binding are coupled. So here we did studies using both the wild type and the dimers which are trapped, which has a disulfide to trap them in the dimeric state. So this is basically the raw data. Okay, so what I'm showing here is the heat release. This is the raw data. This is summarized. The heat release was the stoichiometry. Okay, and this is when we analyze the data, these are the numbers we get. So if we just focus on for our discussion today, the KC binds around 4 micromolar, MIP2 around 25 micromolar. So there's about 6-fold difference in the binding, and the stoichiometry is give or take 2, implying 2 GAGs bind per dimer. Okay, but if you look at the energy differences, I'm sorry, the KD is around like 6-fold, but the enthalpic and entropy contribution, if you see, they're quite different. Okay, so the KC is enthalpically driven. Okay, and remember minus means it's favored, whereas it's entropically disfavored. Okay, so that's the reason it's -7. So it's enthalpically, that means there's most probably, things happening at the reorganization of the side chains and release of water and things of that nature. But the composite is entropically disfavored. So meaning it is more organized in the bound form compared to the free form. Okay. Whereas for MIP2, though the difference here, if you look at it, is only like 1 kilocal, right? The relative distribution between enthalpy and entropy is quite different. It's only like -4, and it is also entropically favored. It's entropically favorable, like -2 kilocals and around -4.3. Okay, so kind of information which you would never ever be able to speculate or guess from looking at the structures of the sequences. So what's the difference between KC and MIP2? So these are the, you can see B3s. So 1, 2, 3, 4 residues are conserved. 4 out of 5. I'm sorry. 1, 2, 3, 4, 5 out of 8 are conserved, and B6 is not there for either of them. So for 2 residues, KC has it, MIP2 doesn't have it. In addition, MIP2 has these 2 residues which are unique to the chemokine, and KC doesn't have it. So this is the combination of these things results in very large differences in the entropy versus enthalpy contribution. Okay, so can we take this to, so we don't have like an NMR or crystal structure. So what we have is structural models. So the way we get structural models is using solution NMR. Okay, basically where you titrate the GAG into the protein. For people who don't know NMR, like basically each peak corresponds to a specific amino acid, which is a change in chemical shift on binding, for example, you can see there's a change, implies either that particular residue is involved in direct binding or it's in the proximity of the binding surface. You can see a lot of changes here, residues over here, some over here, some over here. Blue are the negatively charged residues and red are like non-charged residues. So if you paint them on the structure, okay, so we already know the stoichiometry is 2, right? So basically, this is the dynamic structure. So you have one GAG on this side of the protein and the other GAG on the other side of the protein. So I'm not showing the other monomer for clarity, the other GAG on this side for clarity. So if we zoom in the binding domain, you can see the interactions of all the GAG residues implicated in binding. Once again, the same thing I'm simplifying here, just showing the positive charge residues like R17, K61, 65, 69, and things over here. And once again, the same as here, but here showing the topology, the electrostatic distribution. Okay, here we can see the role of R17 much better in this schematic compared to this schematic. Okay, but once again, this basically is just a structural model. This doesn't tell us whether all these residues are contributing in a similar fashion. If you remember, like if the binding energy is 8 kilocalories or whatever, what exactly is the contribution of each and every individual amino acid? So this is where, okay, so so let me go back, step back a minute. So whatever I said was for MIP2. So now let's look for KC. We did the same thing. We have like a structural model. And so to understand how these residues are doing what they're doing, we did MD simulations, Dr. Desai. And it's what I'm showing here is the hydrogen bonding properties. Okay, the higher the occupancy, the stronger the hydrogen bond. Okay, so here is for MIP2 and KC. This is a direct hydrogen bond, and this is mediated by water. Okay, so you can see right away, with lack of time, I'm not going to go into the details, that first of all, for a given chemokine, all residues don't contribute equally to the binding. Okay, so for example, here the MIP2, okay, contributes more heavily than I'm sorry, these residues contribute more than these residues. And the hydrogen bonding mediated by water, for example, you can see lysine 62 is very dominant, but none at all over here. And the same thing goes to other chemokines. And but actually, that's once again hydrogen bonding lifetime. But so we can convert that into energies. Once again, we can see right away the MIP2, these residues, these two, arginine 17 and K21, show the highest energy, whereas the corresponding helical residues show less contribution, whereas for this protein, corresponding helical residues show higher binding affinity. Okay, so the reason why this is interesting and important is also the fact that historically, the helices were what was thought was important for GAG interactions. I mean, these data show, especially for like MIP2, these end-loop residues, which is another part of the protein, contributing more towards the binding. And but once again, the binding energy between these two is just 1 kilocal, but the topology and the distribution, as you can see, are very different. Also, if you look at the scale, here goes from 0 to -7, here goes from 0 to -11. Like the K62 is actually in question. It plays the most prominent role for this and these two residues, whereas it's sort of more evenly distributed. Okay, so we also did some studies with using polymeric heparin. Okay, so I'm not going to spend too much time here. Anyway, the bottom line here is that we can get this enthalpy entropy issues, and we can also, like doing experiments as a function of ionic strength, we can get insights into non-ionic interactions. Okay, so-called packing interactions, which is quite substantial, like -6 kilocals, and we can also get the relationship between enthalpy and entropy. What is uses enthalpy and entropy compensation, and so kind of issues if you're really interested in the details and from the drug design perspective and things of that nature. Okay, so yeah, this kind of data also shows that when you do it as a function of temperature, and there's burial of polar residues at the binding interface. What I mentioned with delta AP. And a lot of the times you're not interested in this much details on it, but I think you are, and you will be if objective is to understand the mechanistic insights and coming back to the so-called drug discovery process. All right, okay, so I think it basically summarizes whatever I have said so far, and I'll stop there for the first part of the talk, so where we sort of talked about mostly structures, different residues and the thermodynamics, and so like after this I'll sort of go into the kinetics and the molecular dynamics. Any questions?
Yes, I have a question.
Yeah?
On slide 26, when we describe the differences in binding with Kc and MIP2, the Gibbs free energy for Kc, even though it is not entropically favored, the Gibbs free energy is lower. So does that mean that it's a better binding than MIP2?
Okay, so if you purely take in terms of binding affinity, it does bind with higher affinity, right? It's like 6-fold. But see, the delta G is like less than 1 kilocalorie per mole. So if you go purely by affinity, yes, Kc binds with higher affinity. And like in the next part of the talk, I will show experimental evidence. Higher affinity doesn't mean it's better. Okay, so from an in vivo physiology, the affinity has to be correct in terms of what they do, why they do. Did I answer your question?
Yes, thank you.
Okay, all right.
I had a question too on slide 20. So, I mean, you don't have to go too far into it, but I'm just looking at like the top equation. So when you actually do the titration and you get, however much heat was released upon binding. So what are these like? What are one of these different like? I'm assuming they're constants, but you have like PT, Ka, and then XT. Like I think I know what Ka is. I'm assuming that's the equilibrium constant. But
Okay, all right. Okay, so let me see if I can recollect what they are all. So am I going the right way? Okay, all right. So yeah, the PT is the total protein constant, the amount of protein you have, and V is the volume, delta H is the enthalpy, and XT is the amount you are adding at every single increment. So like when you're actually fitting the data, you don't see any of it. You fit it to like a 1:1 equation or whatever, and it spits out the numbers. Yeah, PT is the total protein, N is the stoichiometry, and K is the equilibrium constant.
okay.
Yeah, and I think XT is the variable for every single titration. Like I think maybe it's the relative ratio between the GAG and the protein. I think I'm not sure about the X part.
Gotcha.
Yeah.
All right.
That's
Yeah, I'll
variable.
go.
Okay. So are we good?
Yes. Thank
Okay,
you.
all right. So let's take a 5-minute break, and then we can start the second class.
Sounds good.
Understand, you think, are the ionic interactions, and the ionic interactions, as I said, are predominantly mediated by the lysines and the arginines, and there is also published evidence, some evidence on this. Okay, so in the past I showed you the lysine arginine side chain, and depending because of the very nature of the differences in the side chain, right? Like an NH3 group versus the guanidinium group, the binding interactions between the GAG sulfates and the carboxylate and the guanidinium or the NH3 group is going to be different. right, in terms of the affinity, the lifetime, and so on and so forth. So how do we measure? How can we gain insights into this? Okay, so we will take an example of a specific chemokine. For example, here is a chemokine called interleukin 8 or CXCL8, and this is how the charge distribution looks like. So if you look at the binding site, a lot of the residues, as you can see, is confined to one part of the protein. We already saw an example, and you can see it's a mixture of there's one histidine here, a couple of lysines, one or two arginines. So what exactly are they doing? Why are they doing? Okay, so experimentally, okay, remember experimentally, how can we characterize the individual lysines or the arginines? Okay, this is actually a question for you guys. Okay, from whatever you know, you are interested in a particular lysine, a bunch of lysines or arginines. You really want to know what they do, how they do, and how exactly are you going to go trying to find the answer for this question? I'm waiting.
Sorry, we were muted.
Can you hear us now?
And now I can.
Okay. Can you mutate those residues?
Yeah, my question was, are you asking me to repeat the question?
Yes, please.
Yes, please.
Yeah, okay. So my question to you is, you're working on a GAG binding protein which has a bunch of lysines and arginines at the binding interface. How exactly are you going to go about trying to understand how these lysines and arginines mediate binding?
I mean,
What can you think of which will answer you this question?
I mean, if you have like just like you're showing here, if you do like protein NMR, and if you just have, say, the protein that you think binds to the GAG by itself, and then you analyze that, you get all your chemical shifts, and then when you analyze another solution and you add the GAG, if you see any particular signals shift their position, then that's indicating that they're probably involved in binding.
Right. Okay. So yeah, the data I showed you. Okay, that is okay. In terms of a technique, that was one technique. Yeah. Can you guys think of some other technique?
Can you also mutate those residues?
Yeah, you can do that. Okay, so you can mutate that particular residue, and from a change in the binding affinity, you can infer whether something is, it's involved in binding or not. Yeah. Is anything else? Not really. Yes, I think that's something I really want to emphasize here. Okay, I mean, you can do mutagenesis. And it takes time, right? And depending upon the protein and how if that particular lysine arginine should result in substantial difference, and at least like tenfold to be able to measure reliably. I said that maybe even like 2 or 3-fold, depending upon the technique, you may be able to pick it up. So the challenge, the solution NMR is sensitive. Right, the data which we saw before, like the first part of the talk, what we looked at was a backbone amide chemical shifts. Okay, so in other words, this is the shift we were looking at, but the binding interactions happen right here, right? So the binding interactions is removed by 1, 2, 3, 4, 5 bonds. Okay, so the changes have to be, nice thing about NMR, it is sensitive. And the change in chemical shift could be due to direct binding interactions and also due to indirect, if you're like in the proximity of the binding pocket. Okay, so I'll sort of give you some examples. So the backbone shifts are quite good, but it is not unambiguous. Okay, so here what I'm looking at is I'm actually looking at the side chain chemical shift. Okay, this particular shift. This is the guanidinium group. So these hydrogens rapidly exchange. So we can't observe them, but we can observe these. Okay, so these, for people who know NMR, its chemical shift is different from the backbone amides. So here I'm showing. So what the data I'm showing here is, we are trying to understand. Okay, so what I did not talk about this earlier. So when I showed you the sequences, what stands out for this family of proteins is a lot of the lysines are conserved. Okay, so if a particular residue is conserved, for example, we're talking about lysines or arginines, right? Why is lysines conserved? And also we know from literature, from protein DNA kind of studies, that arginines in general, in terms of mediating ionic interaction, they impart higher binding affinity. So you would think if arginines impart binding affinity, if anything, arginines would be more prevalent or more highly conserved if the binding affinity has to be higher. If since it's the other way around, that as far as the chemokines go, the GAG binding residues, it prefers to have lysines at the binding site. Okay, so the question is why? Okay, both of them are involved in ionic interactions. So what we did here was trying to see if we can get a handle on this by doing metagenesis. So what we did was, there are this particular chemokine has 5 arginines. You can see 26, 66, and 68, and 47. And we know from functional studies, 60, 68, and 47 are involved in binding. And sure enough, okay, you can see on binding there is very little chemical change of these 2 arginines, but there's quite a bit of change for these 3. And so we mutated like 3 lysines. You can see K64, K20, and this is it's another double mutant, K64, K67. And so here by doing NMR, we could definitely show the lysine to an arginine mutation, did not affect the structure, and that these arginines are also involved in binding. So at this stage, it's purely, only thing I can tell you is involved in binding. I can't tell you anything more than that. And so we actually, from the NMR titrations, we can get the binding affinity. Okay, and you can see this double mutant binds almost like 25-fold with higher affinity, like less than 1 micromolar. The wild type is like 25 micromolar, and this particular mutant is, even this one is, this double mutant is also less than micromolar. This is only about a 2-fold difference. So clearly, this is, as we expected from textbook, we substitute an arginine into a lysine. The binding is, it does bind with higher affinity, okay? But nature doesn't want arginines, right? It wants lysines at this position. The question is why? Okay, so we also did some MD to find out what exactly the consequence of the lysines and the arginines. Okay, like for example, this K64, like 10 nanoseconds, right? It engages 2 different the GAG chains. Okay, you can see it's for this particular glycan, and this is an iodo whatever, and a particular lysine, for example, K20 is engages in different side chains. I'm sorry, like a given GAG side chain, for example, it engages histidine 18, here it engages K20. So like in a nanosecond time scale, these interactions are happening. So clearly there could be differences between lysines and the arginines. And so we did this, okay, there's way too much information, I'm going to spare the details. And the bottom line was we did see differences, okay, but really it was very hard for us to make any sense other than the fact that arginine interactions tend to be a bit more long-lived. But lo and behold, what we're looking for really happened when we went for functional studies. When we took these mutants in a mouse, because these chemokines recruit neutrophils, see all the double mutants which binds GAG with much higher affinity, they recruited much fewer neutrophils. They're telling us that higher affinity doesn't translate to higher function. So to do the GAG interactions, I'm going to spare you the details of how chemokines do what they do, but clearly GAG interactions regulate function, and if you mutate to an ALA, right, which reduces affinity, that also results in reduced recruitment. So lower GAG affinity reduces function, higher GAG interactions reduces function too. So that's sort of once again emphasizing the importance of, so they are involved in ionic interactions, but lysines and arginines are definitely not equivalent. All right, so now I'll switch gears and talk about kinetics. Okay, so once again, for people who think about drugs and drug discovery, okay, just like I was talking about enthalpy and entropy, the kinetic constants also makes a difference. Okay, the kinetic constant basically meaning the on constant and the off constant, which is basically your KD. The association constant is ratio of one over the other. So if you can increase the on constant or reduce the off constant, you basically increase the affinity, right? So how do GAGs bind? What exactly are the on constants and the off constant? Okay, so that's basically what kinetics is all about. And as most of you know, and I also alluded earlier in my the first part of the talk. So the technique we use, the only act on really is SPR, so-called surface plasma on resonance, where we basically immobilize the GAG. In the sense, you're also mimicking in vivo function, because GAGs are immobilized as part of proteoglycans and on top of endothelium or epithelium or extracellular matrix. Okay, so SPR can tell us how fast, how slow, how strong, how much, what is interaction. So you can get, this information content is pretty high. You can get specificity, affinity, and things of that nature. Okay, so this sort of just like for ITC, you can use SPR to study binding of anything to anything, like protein to protein, protein to sugars, bacteria, protein to small molecule, depending upon the readout. So what we are interested in is like carbohydrates and the proteins. And so it's I'll spare the details. Basically, you measure this in real time, just like in ITC, where the readout was change in the heat. Here, the readout was due to a diffraction, the way the system is set up, due to change in refraction. You are able to measure binding. Okay, so as I said, we collect data in real time, where we mobilize the GAG and add the protein. And it's unlike ITC, one advantage of SPR is you need less protein, and also you can repeat the experiment. You immobilize it on a chip, so once you use one protein or one concentration or whatever it is, you can wash it off and use the same chip over and over and again for quite a few times. And so you sort of went through that. So this is how a profile look. Okay, so this, for example, this is your GAG and that's your protein. I think the actual example shows mostly is an antibody. So this is the association. Okay, so once you stop adding, it starts dissociating. Okay, so this the nature of the profiles is directly proportional to the kinetics. Of course, the height is proportional to the concentration amount you add. You do this like on different concentrations. I'll show you some later. And then once the experiment is done, you regenerate and start all over. Okay, so it's fairly simple conceptually. You can sort of get a big picture of why we are doing what we are doing and what we are getting.
What is on the y-axis in that graph?
I'm sorry, the y-axis is just a readout of the extent of binding. It's an arbitrary scale. The more protein you add, it basically, A is binding to B, right? You keep seeing a signal until the binding saturates. This sort of gives you the on constant, right? And that gives you the off constant once you stop the addition.
Thank you.
All right. Yes, we already talked about this. So there are limitations. Okay, so like sometimes the concentrations can be too high. There's a mass transport limitations mathematically involved, and the optimization could be an issue. Like especially things like when the system is bad and it's like precipitation and things of that nature. And a lot of the times, with respect to any technique, ideally you want a 1:1 binding, right? And definitely GAGs and proteins, the binding is not 1:1. And so you may have to fiddle around and fool around and sort of understand what the system is and accordingly interpret the data. And the binding constants we get generally is like an apparent binding constant. It's not a real equilibrium constant. But something to remember. And also I'll show some data so we can talk about it. Okay, so now we'll take a real-life example of a system we have studied in the lab. So this particular chemokine, actually it's, has like 2 binding sites. Okay, so what I'm, in a dimer, which I'm showing, like it's a 1 binding interface. You can see like 5 residues from one monomer and 5 residues from the other monomer, constitute a binding interface. The GAG runs along like this. And the other phase, you have like 4, bunch of residues. So the binding interface is extremely extensive for this particular chemokine. Basically, your chemokine is sandwiched between 2 GAGs and involving very many different interactions. So the question is, what exactly are the role of these lysines and arginines? How much are they contributing to the binding affinities? So here is an example of the data. Okay, so like the red is, the black is the raw data. So with each is like with increasing concentration of the protein, you can see the binding is on constant very fast, off constant is very fast. And this is fitting the data to the on constant and equilibrium constant. And so this is like from one of the mutants. This for the wild type. You can see right away the binding is much weaker. You can see one of the plots where we take the concentration as a function of response. You can see a typical dose response curve. And here we can sort of plotting the relative binding affinities. If we take the wild type, you can see the mutants from both alpha domain and the beta domain result in lower affinity, not a big difference between lysines. There's like 1 or 2 arginines here. In fact, the arginines seem to result in much lower compared to some of the lysines. So this is sort of tells you that both the domains are involved in binding, but what exactly are the kinetics? Okay, so a lot of data here where I'm showing you the on constant and the off constant for the individual mutants. Okay. So you can see like the on constant is generally for any protein is what is called diffusion control. It tends to be like 10 power 4 or 10 power 6. And you can see all the numbers are not very different. Okay, so around 2, 1, 3 kind of thing. 2, 1 or 2, 4. Whereas the off constant, the 0.02. Okay, the lower the numbers, lower the diffusion, lower the off constant. And almost for all the mutants, they are much higher, like 0.02, 0.16. And the off constant is coming off 8 times faster. And you can see the KD is about 8-fold the weaker. Okay, so 1 micromolar versus 8 micromolar. So saying that kinetically the GAG interactions are diffusion, the on constants are diffusion control, but the off constant is what dictates the change in the binding affinity. So as I said, like from a drug discovery perspective, you can see, right, what I said is that you want the off constant to be as low as possible. So this sort of tells you if you can increase the affinity, it seems, for the way to do it is not as much as on the on constant, but to minimize the off constant, whatever the off constant once again is dictated by the interaction and the enthalpy and the entropy, right? The reorganization of the binding interface and so on and so forth. But the objective is to show you the structural insights and how is it coupled to the actual properties which dictate the binding, like kinetics and thermodynamics and the binding interactions. Okay, so so this is just in vitro. So the fact that they bind with lower affinity doesn't mean, what does it mean in terms of function? So like at the end of the day, you want to validate that these interactions are relevant. Indeed, what you see is that once again, like in the mouse model, all of the mutants are less active. Like this particular mutant is significantly less active because it's also resulted in receptor interactions. Proof of concept that both domains are involved in binding and a bunch of residues from both monomers of the dimer are also involved in interactions. Okay, all right. So we just talked about, so coming back to IL-8, so CXCL8, we talked about CXCL1. We already told you these residues are involved in binding. So here now I'm going to go a little bit more into the details. Okay, so like this sort of a pushing the envelope kind of experiments. Okay, so like initially I showed you this arginines, right? how these arginines are involved in binding, and we saw like 68, 47, and 69 involved in binding. But what about its properties, like the dynamical properties? So we can measure dynamics, using NMR. There's a property called heteronuclear NOE. If it's like goes from -1 to 1, if it's like minus, that means these residues are dynamic. 1 means it's much less dynamic. Like R6, we knew it's an N-terminus, which is unstructured. So R6 being unstructured or being dynamic was not surprising. But R60, which is very much part of the binding interface, is more dynamic compared to like 47 and 68. Okay, so this is the kind of information which you cannot get from the structure or from binding interactions. So you need to measure dynamics, because these experiments measure directly the dynamics of the side chain. Remember, what I'm looking at is the N epsilon H group and not the backbone MI. So this tells you that this the different residues in the binding interface have differential dynamic characteristics. And so if you look at so this is called NOE, and other relaxation parameters you can measure is also called T1 and T2. And the T2, you can see like these two residues correspond to R6 and the 60. The T2 also tells you that the dynamic characteristics are very different from the other two GAG binding residues. The T1 is sort of very similar. So this is for arginines, and the lysines, once again, kind of information which is very hard to come by. I think we are the first people. We had a paper which came out last year where we have actually characterized the lysine side chains in terms of what we can learn in terms of its binding interactions. So I'll sort of come to that soon. So here, once again, we are looking at the dynamics of the lysines. So like compared to the arginines, okay, this is actually in the bound form. Because in the free form you don't see any of the signals, and you can see the NOEs are all negative. Even in the bound form, it's highly negative, all the way up to -3. All of them, doesn't matter whether that lysine is involved in binding or not. It tells you the lysines, even in the bound form, are highly dynamic. Because remember, I talked about enthalpically and entropically. So the lysines are sort of involved. Seems like interactions are both enthalpically and entropically favored, because you still have ionic interactions with this enthalpically favored, and but still in the bound form they're dynamic, which is entropically favored. Okay, so we also see some differences in T1s and the T2s. So once again, these are not easy experiments, but if you can do it, takes time and effort, but you can learn different kind of details which you're not going to get, experimental insights which you're not going to get from any other technique. Okay, so I just talked about CXL1 earlier. If you remember that this is, I should have shown this slide earlier, that it's sandwiched between two GAGs, so-called alpha domain and the beta domain. And I'm also showing the binding across the alpha domain and the beta domain. So we will this is the backbone chemical shift changes, right? And so here I'm once again using the backbone chemical shift changes. I'm looking at another CXL, another chemokine. It sort of binds in a very different fashion, very similar to KC and MIP2 I talked earlier. We have like two equal binding sites, one binding to the monomer. Unlike there, it was spanning the dimer interface. So here we made use of the lysines. Okay, so I just showed the data for IL-8. So if you look at the lysine side chain in the free form, you see only one lysine, K21 and K41. Okay, basically this tells you this K21 is sort of involved in intramolecular interaction, maybe with an aspartate or glutamate. All of the lysines, I think each one of them has like 8 or 9 lysines. They are, in addition to the GAG binding residues, there are also other lysines. They are exchanging rapidly with the bulk solvent. That's the reason you don't see it. That's because they're very, very dynamic. So what happens is these lysine and the side chain NH3s, once it binds to the GAG, okay, they don't exchange. They're involved in ionic interaction. Because the exchange is substantially lowered, you can see the individual peaks. Okay, and this for a DP8 and this for a DP14, and you can see done the labeling, and so the very fact that we see these peaks tells us that These residues are involved in GAG binding. Okay. And also we also did other spectra, okay, where you look at the hydrogens attached to the adjacent carbon. But remember, I cannot measure which with the hydrogens attached to the adjacent carbon, which don't exchange fast. Okay, you can see, but so you can here I can actually measure the chemical shift change. Okay, so the point I want to make here is here I don't know the chemical shift in the free form, okay, because it's exchanging very fast. But here I know the chemical shift in the free form and in the bond form, from where I can measure the change in binding chemical shift, just like the backbone, and you can see, okay, the change in chemical shift. K71 is not involved in binding at all, whereas its backbone chemical shift changes. Same thing with K76 and the K41, which was showing a peak in the free form, doesn't show a change in chemical shift, implying it's involved only in intramolecular interactions. Whereas K21, which is involved in intramolecular interaction, is also involved in intermolecular interactions. Okay, so from this is the easiest experiment we can do. It's just a backbone. Okay, but doing this so-called HISQ, and H2CN, we could say for sure K29 is not involved in binding. And same thing with K76, it's not involved in binding. So like, the bottom line is HSQC is because of the backbone. Most of the time it's correct, but sometimes it can be wrong. For example, here it did not show a change in chemical shift for K29, but other two techniques showed it's involved in binding. Because of the empirical nature, you could have chemical shift changes which involve binding, and it could be a structural changing which may go in the opposite direction and cancel out each other. Okay, so once again, we are making you get a feel for the dynamics of the lysine side chains, how the dynamics is substantially pinched in the bound form, and also how you can exploit this information to try to understand the role of dynamics. As I said, it's coupled to thermodynamics because entropy is very much involved in the binding process. Though the lysine side chain continue to be dynamic, but the lysine side chain amides and the NH3 group are not exchanging that fast. You get a lot of information, but a lot of ion waving, but still you're learning a lot more. You knew a lot more than what you did before not doing this kind of experiments. Okay, so histidines. Absolutely nothing, as I said, like there are a couple of crystal structures which are shown involved in binding, maybe some mutational studies. There's no direct experimental data, okay, and the one great thing about NMR is, it's explicitly sensitive in chemical shifts. And histidine, just like, unlike other amino acids, we talked about it, because of its side chain, it actually can exist in 3 different forms. So-called, this is, protein biochemistry, so-called imidazolium state, where both the protons are protonated, okay, so it's localized, and so-called the N-epsilon 2 tautomer, where the hydrogen is on the nitrogen, and N-delta 1 tautomer. Okay, so depending upon what state you have, the chemical shift profiles look very different. Okay, so this is how it looks if the histidine exists like this, with the protons on epsilon 2 like this kind of an L and this kind of an L. Okay, so how does it exist in proteins we know? So this is IL-8, CXCL8. You can see both. There are 2 histidines, both are very highly conserved. And we knew from previous experiments H33 is involved in binding. And we can also, by titrating the histidines, you can get the pKa. The pKa is like really low, like less than like 4 to 5 kind of a thing. Whereas if it's like solvent exposed, it's like around 6. So the pKa is substantially lower, indicating these histidines are involved in some kind of intramolecular interactions, or its environment is like more charged or something along those lines. There's something unusual about both the histidines from measuring the pKa. Okay, so this is in the free protein. So what happens when it binds to the GAG? I'm sorry, this is small. So I'm showing the individual spectra when it binds to heparin, when it also binds to the receptor. Both the profile remains the same. the shape, meaning both of them exist even in the bond form as epsilon2. But there are changes in chemical shifts, which you can see when I zoom in, when I plot the free and the bond form, indicating that one of these, H33 is involved in binding to, I'm sorry, the H18 is involved in binding both to heparin and the receptor. Okay, so but when you come to CXCL1, okay, this has never been observed before. Both the histidines in the free form exist in two conformers. Okay, so called imidazolium, you can see the blue and the N epsilon 2 conformer, okay, which I'm showing. We have plotted in different colors so that you can see it, and you can zoomed in this region so you can see the two peaks. Okay. So you have both two histidines, two very similar chemokines, all right, but the state of the histidine section imidazole is very different. In one case, it exists in one conformer, in the other case, it exists in two conformers. So what does it mean? So when you do the binding studies, when it binds to the heparin, the other conformer completely disappears. Only thing we see is the imidazolium state. Okay, and but the H34 is not involved in it's removed from the binding site. Even then, it adopts the same conformer as the histidine, which is in the binding site, implying there's this crosstalk that they're coupled interactions, mediate coupling, direct interactions, and vice versa. Whereas for the receptor bound state, that's not the case. Okay, the H19. So the H19 exists in the epsilon 2 state, and the H34 exists in the imidazolium state. Okay, so so kind of information which you really cannot get anywhere, right? It shows the power of NMR, and most importantly, how two similar chemokines exist. Two different conformers, and how they're binding to the GAG is very different. Okay, so sort of gives you some kind of insight into how in vivo interaction could mediate, affinity, specificity, and function. Okay, so finally, I'll also talk about the polar interactions. As I said, there's absolutely no experimental evidence other than maybe like one or two crystal structures where people sort of imply side chain interactions for asparagines and glutamines, and then Dr. Desai and this previous postdoc, they did some MD kind of studies implying, showing evidence that asparagines and glutamines could be involved in interactions, and they could play a role in specificity. So here I'm showing examples, once again, sort of power of NMR, where we can look at this region of the spectrum. Only residues we see is asparagines and glutamines. Okay, you can see like some changes, and some residues showing a lot of changes, for example, over here, one over here, providing structural evidence. Better than this, I think this here you can see a lot more clear that this asparagines and glutamines also involved in side chain interactions. And most energetically, I'm not sure, we have done some MD stuff with Dr. Desai, and we see that energetics are much, much less than from lysines and arginines, fraction of a kilocalorie. Maybe 1 or 2 could be like 1 kilocalorie, 1.5 kilocalories. Most likely that these residues, the asparagines and glutamines, are also not conserved at all, unlike lysine and arginine. So these lysine and arginines most probably play a role in some kind of specificity. Okay, what exactly is the nature of specificity? I'm not clear at this time. Maybe dictating the geometry or fine-tuning the binding affinity in terms of in vivo function. And I'll stop right here. So I'll take questions. For either of the talks.